Hello everyone, welcome to Zeta Biology. Today we are going to look at nucleosides. It's a topic from Molecular Basis of Inheritance chapter, and it's very important for you to understand it because it is the uh, basics of the entire chapter. So let's get started. So nucleoside are the building blocks of nucleotides. And nucleotides, in turn, are the building blocks of DNA and RNA, that is, the nucleic acids. Okay, so we have nucleoside. So nucleosides are basically made up of nitrogen bases and pentose sugar. Only two components are present in the nucleoside, and the bond present between nitrogenous base and pentose sugar is called N-glycosidic linkage. So this is a really important MCQ, so you should remember it. The bond present between nitrogenous base and sugar is N-glycosidic linkage. So next, uh, so pentose sugar, the first component. Pentose sugar means 5 carbon sugar, and it is present in nucleic acids. Okay, so we know that there are two types of pentose sugar, ribose and deoxyribose. So ribose is present in RNA and deoxyribose is present in DNA. Ribose is different from deoxyribose. Why? Because the carbon number second of ribose sugar carries an extra hydroxyl group, that is an extra OH group, whereas in deoxyribose this OH group is not present, only NH is present on carbon number second. So that is the basic difference in the structure of ribose and deoxyribose sugar. And the second component is nitrogenous bases. So these are glucosamine molecules, and these are of two types, as we know, purines and pyrimidines. So purines are adenine and guanine, and pyrimidines are cytosine, uracil, and thymine. Okay, so that's how you remember it. Pyrimidines are cytosine, uracil, and thymine. Three pyrimidines and two purines. So purines and pyrimidines, my basic difference is that in purines, two rings are present, as you can see. In both adenine and guanine, two rings are present, whereas in pyrimidines, only single ring is present. So these are single ring compounds. Okay. Next, types of nitrogenous bases present in DNA and RNA. Now, as we know in DNA, adenine, thymine, guanine, and cytosine are present, whereas in RNA, adenine, uracil, guanine, and cytosine are present. So, in both of these molecules, the major difference is that thymine is present in DNA, and in place of thymine in RNA, uracil is present. So, uracil in RNA makes it more chemically reactive, and that is why RNA as a whole becomes more chemically reactive than DNA, and that is why it is less stable. Okay, and thymine is basically 5-methyl uracil, and thymine is only present in DNA, whereas cytosine is present in both RNA and DNA. Okay, next, types of nucleosides. So now we know the nitrogenous bases present in DNA and RNA. Now let's look at the nucleosides that are formed, that are present in DNA and RNA. So obviously different nucleosides are formed in DNA and RNA. So when the nitrogenous base adenine is present, it will form the mm, it will form the nucleoside deoxyadenosine in DNA, whereas in RNA it will form adenosine. Same with guanine. In DNA it will form deoxyguanosine, and in RNA it will form guanosine. And cytosine will form deoxycytidine. Remember this because it's not the same. Deoxycytidine in DNA, and in RNA it will form cytidine. And thymine will form deoxythymidine in DNA, whereas in ribonucleoside, that is RNA, it will form uridine. So now you have to remember this. Thymine will not be present in RNA, because uracil is present. So the nucleoside that will form in RNA is uridine, not thymidine. Okay, so remember that. And